welcome to March's Genomics Lite, titled Genomic Cartography. And we're joined by Elena and Feri this afternoon, who are from the Wellcome Sanger Institute, and we'll get to know them a little bit more in just a second. I'll whiz through this first bit because, as we saw, the majority of people had been on a Genomics Lite before. But for those that are new, I'll quickly just let you know a little bit how the session works, what buttons to press if you have any technical issues, etc. All of these events, they aim to help everybody to explore the science of genomics and what it means for our lives. We will be sharing different views, different opinions throughout the session. There'll be polls for people to express, sort of answer different questions, and there'll be a Q&A. So we might see different points of opinion come through. So we just ask that everybody respects any of those different opinions and experiences that might come out from that. And I will moderate this platform and help to ensure that everybody, any technical issues and stuff like that go all smoothly. So if that happens, these are the buttons to press. So essentially, if you have any technical issues today, use the chat button. That will come to me. I will try and help with in the realms of what Zoom will allow me to fix, but I will do my best to try and help. So if you've got any issues with cameras, you can't hear us, anything like that, put in the chat and I will try and sort those out for you. If you would like subtitles with the talk this afternoon, just use the closed caption button, and that will provide a whole subtitled talk option for you. And then, as I say, we've got a Q&A that will be happening. If you have a question, what I just say is put those questions in there from the start. Don't hang on to them to the very end, because there might be an opportunity where we ask them during the session rather than at the very end. So if there is anything that comes to mind, put them in straight in the Q&A and we'll get to them at the right time. So let's do quick introductions. So my name's Sam. I'm a Science Engagement Officer at the Wellcome Sanger Institute. We're based here. This is the Wellcome Genome Campus on a very sunny day. I don't think it's that sunny today, but it's, as you can see, quite a big campus there, all dedicated towards freely open, accessible genomic science, essentially. And we are joined by Elena and Feri, and we'll get to hear a little bit about how they got to working at the Wellcome Sanger Institute. So this is just a quick rundown of how the talk will go, and we'll be wrapped up around about 5:30. And this is being recorded, so if any reason anybody needs to jump off or the internet goes, anything like that, it will be on YouTube, and I'll send an email with the link so you can watch that. Watch it back. Elena, over to you. Tell us a little bit about yourself, how you got to the Wellcome Sanger Institute, and yeah, lovely to have you with us.
Yeah, hi. Thank you so much for having us. So my name is Elena. I'm a postdoc at the Hannover Lab here at the Wellcome Sanger Institute. And basically my journey into academia and science started with a bachelor in molecular medicine in Tübingen in Germany. I was super interested in science already during high school, so this was an obvious choice for me. During my bachelor, I went abroad into the US and into Belgium, where each studied for one semester and kind of studied immunology, infectious diseases, and microbiology. I then came back to Tübingen to do my bachelor thesis. I worked on malaria because I was super interested in kind of saving the world from diseases, and malaria is like a big disease. So after my bachelor thesis, I continued in Tübingen with my master's degree, and my master thesis was then in the hematopoietic system, and I looked at stem cell development from human cord blood samples. I then decided to do a PhD in immunology. I went to LMU Munich in Germany for that, and there I worked on infectious diseases mostly. So I looked at the immune response to infectious diseases, And I used yellow fever vaccine virus, for example, which is a live attenuated vaccine virus where you can basically vaccinate people, and at the same time you're infecting them with the virus, and you can monitor how they react to this vaccine virus over time. And it's super safe for them, and very, very good if you want to see like what happens at the beginning of an infection. Then COVID hit. I worked on SARS-CoV-2 infection as well. And then I decided to do a postdoc abroad, and I decided to come to the Hanifa Lab here at the Wellcome Sanger Institute in Cambridge, UK. And here I'm working still on immunology, but more developmental immunology. And I'm now focusing more on the computational part of the science. So I kind of stepped from the wet lab in the direction of computational analysis more now in this position.
Amazing. And how long have you been with us at the Wellcome Sanger Institute?
Now almost a year.
Okay, amazing. And because I know we've got lots of content to go through, so we can have a little bit more of a chat about some of this later on in the Q&A as well. But Feri, how about yourself?
Hi, everyone. So my journey into science, it started when I was in middle school. DNA, the term, caught my attention during a biology class. And so I'm from Iran. Then later on, when I was in high school, I decided to move countries to study more about genetics and understand all of the fascinating things in this field. So I did a bachelor's degree in medical genetics at Swansea University. And that was, those 3 years was quite productive. So I also did a summer internship, which I researched repurposing of an existing FDA-approved antifungal drug for treating pancreatic cancer. And in parallel to my scientific journey, I also worked as a call handler at the ambulance service during COVID-19 pandemic. And I was also a student ambassador at the university. Then after finishing this journey, I was very excited to be able to apply all of those knowledge that I've gained into kind of wet lab. So I did a research placement at the Winheim Institute. So I came into Cambridge area, and that was where I got introduced to world of epigenetics and stem cells. And after that, I joined Sanger Institute to study blood immune cells using cutting-edge single-cell technologies. And after that, I joined the Hanifa Lab as an advanced research assistant. And now I'm mainly focusing on skin development, stem cells, genomics, so combining all of the fields that I've been excited about for a few years now.
Amazing. And how long have you been with us at the Sanger so far?
In total, so over 2 years.
Over 2 years. Amazing. Well, it's lovely to have you both with us this afternoon, and I can't wait to get started. So I'm going to stop sharing and hand over to both of you. So yeah, let's do that. Hopefully the technology all works now. Perfect.
Can you see the screen? Yes.
Yeah.
Good. So great. So we are going to talk today about genomic cartography and the generation of a human cell atlas. And basically, I'll start with the outline. So I'll introduce you shortly into the work that we're doing here in the Hanifa Lab at the Wellcome Sanger Institute. Then I'll give you a short background in transcriptomics. Then Feri will take over and explain to you how we use the human cell atlas, in particular the skin atlas, to answer some interesting scientific questions. And then we'll give you a perspective into spatial transcriptomics. And basically, here at the Hanifa Lab, we are located at the Wellcome Sanger Institute, as already mentioned, and you can see the group here. having fun outside on the campus, playing Jenga and eating cake. However, this is not all we do. We mostly concentrate on the science, and we are super interested in the human development and in kind of understanding how the human immune system develops over time, from early time points in the embryo to late time points and also through adulthood. And we've published quite a few papers on this, on embryonic development. So for example, one recent paper was on the yolk sac, which is like the first place where immune cells can be formed. And then we also published about liver, bone marrow, and some different organs in the human embryo. And we are also contributing to a whole embryo atlas, where we're kind of looking not only at the different organs, but like at how it's all connected together. And we're trying to use this data to understand how the human develops, how diseases develop, and to use this knowledge, for example, to establish organoids, like a yolk sac organoid, and to improve skin organoids. Now, the Hanifa Lab is part of the Human Cell Atlas initiative, and the Human Cell Atlas is basically a massive collaboration between different institutes, laboratories, and research groups that aims to create a complete map of all human cells in the human body in order to understand human health, diagnose, monitor, and treat disease. And the Hanifa Lab is only one part of this. So here you can see a recent screenshot I took from the Human Cell Atlas website, where you can kind of see how many countries are actually involved in this massive project. And the human body has like 30 to 40 trillion cells. So there's really a massive work effort that a lot of people are contributing to. Now I'll give you a background into transcriptomics, because this is one of the main methods we are using to generate the Human Cell Atlas data. And basically, all human bodies are made up of a lot of tiny units, which are the cells. And here you can see a picture of human skin, where you can kind of see how different cells come together to form a tissue. So like a brick wall is formed out of small building bricks, the human body is formed out of multiple cells and multiple different cells that have different morphology and different functions. And these different cells make up the tissue, which makes up the organ system, and the organism. So if we understand these small units and how they're connected together, in the end, we will understand the whole human body. Now, each individual cell contains genetic information in the form of DNA, and you might have heard this before, but I'll just repeat it anyways. And this DNA is in the form of the double-stranded helix in the nucleus. And basically, almost every cell in the human body shares the same genome. There are some exceptions where some cells have kind of no nucleus, but basically the genomic information is the same in all the cells. And this genomic information is then used to produce mRNA. And the mRNA is different between different cell types. And the mRNA then is translated into protein, which is then also different between different cell types. So basically, by looking at the transcriptome and at the proteome, you can see what makes a cell unique. And this is basically the central dogma of molecular biology, where DNA makes RNA, RNA makes protein. And while all the cell types have the same DNA, they have different RNA and different protein. And what we're measuring with transcriptomics is basically the RNA in each individual cell. So we are doing RNA sequencing here. There's also a way of measuring the DNA, like DNA sequencing and the protein, but we are mostly focusing on the RNA sequencing part. Now, RNA sequencing is a way of measuring the RNA in a sample. So in the past, what was done usually is you took a piece of tissue, like for example, this piece of skin, you isolated the RNA out of this tissue, and then you used this to generate sequencing libraries. You sequenced this. That meant that you kind of read what kind of mRNA you have in the sample. And this then led to a lot of information. However, since all those cells were mixed up, you couldn't say like which cell type contributed what to your readout. So you couldn't say like these cells up here might have a different mRNA as other cells. So now a lot of people are using single-cell RNA sequencing instead of bulk RNA sequencing, where you're looking at individual cells and not at the whole tissue. And this enables you then to look at what cell expresses which gene and kind of differentiate between all the different cell types that you have in the tissue. And an analogy that is often used to kind of explain why this is so important is the fruit salad analogy. So if you imagine that the tissue is like a fruit salad, like it contains different cells, or the fruit salad contains different pieces of fruit like banana, apple, kiwi, bulk RNA sequencing would be like putting this into the mixer and making a smoothie, which means that you will have some nice output, like you will taste maybe banana and apple, but you will not be able to taste like the smaller parts of the smoothie, like the kiwi or something like this. And you will not be able to really say like what contributed to the taste that you have. And single-cell RNA sequencing is basically looking at all the fruit pieces by itself and kind of getting the gene expression of each individual cell. And this might even enable you to see differences between different parts of the apple. So this will give you like a really good insight of what is happening in the human body. Now, in the last 15 years, single-cell sequencing technologies have advanced greatly. So it all started in 2009, where a single cell was actually sequenced. And the early techniques of single-cell sequencing were basically based on you putting a cell into a tube or into a well, and then generating libraries from each individual well. So I started, for example, using SmartSeq version 2, where I had like 3 96 well plates, and in each plate, in each well, you had to pipette and generate your libraries. And nowadays, what we are doing mostly is droplet-based sequencing, where you don't have to do the separation of the single cells, but basically you're generating droplets that then contain a single cell by an oil emulsion. And this enables you to really scale up this and to now sequence millions of cells and even profile the whole organ or tissue, and therefore, in the end, the whole organism. And there are still some restrictions as to how many cells you can profile. So for example, one restriction would be money. This is an expensive technique. So 10,000 cells costs about 1,700 pounds. And another restriction would be the tissue availability. So for example, we are working with developmental studies and with very rare tissue that comes, for example, from human embryos. And so this is like a limiting factor. So this leads us to our first poll.
Amazing. So yeah, so we've got a poll, and what I will also do after this poll, we've had a question come in, so I think we'll answer that question now as well. So first of all, the poll is, and I'll launch it now for everyone, which is, there we go. Let me launch that for you. So this says, how many cells do you think were profiled in the last prenatal skin single-cell RNA study by the Hanifa Lab? So we've got 44,000 cells, 440,000 cells, 4.4 million cells, or 44 million cells. I think I've said those those right, those shifts right. So yeah, have a quick vote, see what you think. And yeah, we'll get the answer across as well. So if you haven't put in an answer but would like to do so, please do now. If not, I will close the poll now. So we have got, the majority are saying 4.4 million cells. But we've got a spread across all of the answers. So what is the answer?
Okay, so the answer, actually, you're overestimating the amount of cells a little bit. So we sequenced 440,000 cells. And as I said, the sample material we get is quite rare and precious. So it's we are not able to sadly get as many sample materials as we would need for 4.4 million cells. But it's a good estimate, and there are studies that are sequencing this many cells.
Okay, just before we move on, if that's all right, there was a question that came in which was just around the types of RNA, and I just thought it might be good to maybe just pause quickly now whilst we're at that point. It's a great point just to raise. Keep throwing in any questions that you have into that Q&A button, and we'll try and get them at appropriate. So the question was, it's from a teacher, was saying that I know that there are at least two types of RNA, mRNA and tRNA, but if you are saying that different cells have different RNA, then they were saying that they suspect the story that they tell at GCSE and A level is a little bit of an oversimplification. Can you potentially expand upon that in any way?
So what we're measuring with transcriptomics is the mRNA, and basically each individual cell produces reads different parts of the DNA to produce different mRNA. And this is basically what I meant with the different cells have different mRNA. So a lot of them have also like different types of RNA within the cells. So that is also a factor. But that's not exactly what we're measuring with the transcriptomics part.
Okay, super. If there are still if there's still a little bit more to that question, do just put into the Q&A and we can get to it a little bit later as well. Okay, perfect. Back over to you, Elena.
Great. So now we profiled in the last study 440,000 cells, and what does that mean in the data that we get back from these studies? So basically, when we're talking about a single cell sequencing, this means that for 440,000 cells, we get the information of all these different genes that are expressed within that individual cell. And depending on which human genome you align this to, this is a little bit different, but we have, for example, 36,000 gene expressions measured here. So that means that we get a matrix of 440,000 times 36,601 in the end after the sequencing, which is massive. So if you imagine how to interpret this data, you could, for example, now plot each individual cell and each individual gene expression against each other, and then you would have like a massive plot that nobody wants to look at and that nobody understands. So one important thing is trying to visualize and make sense of this data. And of course, we could do interpretive dancing to make sense of the data, but this is usually not accepted in the scientific community. So what we've decided on doing is to reduce the dimensionalities. And reducing dimensionalities basically means to turn this matrix into something that we can understand. And we're reducing the dimensionalities by putting this into a two-dimensional space that you can see here, where basically we are summarizing the information that we get from this matrix and plotting this into a two-dimensional space. So, for example, we are putting cells that have similar gene expression next to each other, for example, down here, and some that are different from them on the other side of the plot. So basically, this is a method of reducing dimensionality, or like turning matrices into colorful blobs. Now, there are different methods of doing this. So here you can see, for example, UMAP and TSNE. That's basically just different ways of doing it. And the idea is that we are reducing the dimensionalities from these massive matrices into two-dimensional spaces by grouping the cells by their similarity. Then we are kind of giving them a name. So we are kind of giving them a group name. So for example, cluster 1 and cluster 2, cluster 3. And then we are trying to give them a more meaningful name, which means we are trying to annotate them. And we do this by comparing or by looking at the gene expression of the different clusters. And first, we can compare this to literature. So for example, when we see that one of our clusters or groups expresses CD3E in a very high way, we can compare this to literature, and it is known that this is found on T cells. So we can say, oh, this is most likely a group of T cells down here. And similar with other genes that are expressed by the cells. And that way we can kind of give those names that are meaningful and that we can work with. And if we find a cluster of cells that hasn't been described before, like the gene expression is completely different from what is found in literature, we can actually just give it a new name and kind of call it by the gene expression we find, or give it a funny name, whatever people prefer. Now, once we've kind of grouped these single cells into similar cell groups, we can then ask biological questions. For example, like how many of those cells do we have in the tissue? How are they different between a healthy patient, between a diseased patient? How do they change over time? So for example, when we're looking at development, like how are they changing during the aging process and so on. And a lot of these data sets that are generated by single-cell sequencing are then combined into a human cell atlas, where we can kind of look at the different tissues at the different ages of healthy patients, of diseased patients. And this information can be used for therapeutic screening. We can use this to look at how different are humans between each other, so human genetic variation. And we can also use this to kind of improve organoid models or compare to model and non-model organisms, like, for example, compare to the mouse and see how different mice are from humans. And with this, we come to the second poll.
Amazing. Yeah. So as you mentioned before, so I'll launch this as well quickly so that people can start voting and reading it as well. So I'll read it out. So as part of scientific research, human embryos are sometimes used to study human development through the single cell RNA process. How do people feel in the room around this use of human material? So have a think about this. You don't need to, if you don't want to vote, don't feel like you need to, but it's just, we're conscious that, you know, in all of the work that we do, it's very important to think about societal and ethical implications on all of this stuff. So it's great to hear potentially what the opinions are of our audience as well on these issues. So if you'd like to vote on how they feel, you could, there's option to supportive, neutral, concerned, or unsure. So have a little think and put your answer in if you'd like to. I'll give maybe 5 more seconds. So if you haven't put in an answer and would like to, please do so now. If not, I will end the poll. Majority in the room are supportive, but again, we've got a mix of neutral and also concerned. So yeah, it might be, if we have time in the Q&A, it might be really interesting to talk about maybe some of those ethical considerations that you have to go through as well.
Yeah, that sounds great, actually. So now I'll hand over to Feri, who will actually show some examples of how you can use the human cell atlas, or how you can use especially the skin atlas to answer biological questions.
Hi, everyone. Thank you so much to Elena for giving a really nice background to the human cell atlas and the analysis, which is usually the complex part. Yeah, before we dive into the skin atlas, I think let's start with a poll question about skin.
Amazing. Yeah, so this is another one of those numbers ones. So how many cells are in 1 square inch of skin, do we think? So let me launch that quickly now for everybody to quickly have a vote on. See where people are lying on this one. So far off the mark, we've got a real spread across all of the answers so far. So it'll be interesting. So yeah, 1,900 cells, 19,000 cells, 1.9 million cells, and 19 million cells. So how many cells do we think are in 1 tiny little square inch of skin? Okay, if you'd like to put in your answer and haven't done so, do so now. If not, I will end the poll in just a second. Okay, so we've got a real spread. The most popular vote was 19,000, but 1.9 million and 19 million were also very popular. So, Feri, tell us what the right answer is.
Okay, so sorry, I don't know.
You had anticipation.
Yeah. So I think a lot of people might have underestimated how complex the skin and how multicellular, cell-rich the skin is. So yeah, the answer was 19 million, which is quite a lot of cells for 1 square inch. So I hope this has kind of captured everyone's attention to skin, our favorite organ for today. So what is cool about human skin? First of all, human skin is the largest organ in our body, and in fact, it accounts for 10 to 15% of our body weight. And how does it develop with, all of these different cell types? So it develops during development, embryonic development, and it continues until birth. So it's like a very step-by-step process, layer by layer, and new cell types start appearing. And at birth, the skin has 3 main layers. So we can see in this image, we have epidermis, the outermost layer of the skin. Just under that, we have the dermis, and we have hypodermis. But in addition to these main layers, we have different structures and substructures. So 2 of the main substructures of the skin are hair follicles and glands. So we have different glands. And in addition to that, we have nerve cells and fat. So despite skin being one single organ, it has a lot of variability in terms of features and functions. And so let's take a closer look at what these variabilities could be. So if we look at the spatial aspect of skin, so what does it mean? So skin from different areas of our body, they're very different. We know that on our scalp, we grow a lot of hair, but on the palms of hands, we do not have any hair. The temporal aspect basically captures the aging. So we know that the skin keeps changing and throughout aging. The gender aspect, it captures the fact that we know there are a lot of differences that are mainly hormone dependent between different genders. And the ancestral origin of the skin can also have an impact on specific features of the skin color, and as well as that, certain diseases we know could be more prevalent in specific ethnicities. And lastly, wound response. So the skin, especially adult skin, once it is wounded or damaged, it develops scar tissue. And that tissue is completely different to a healthy, unwounded skin. So I hope we can all appreciate how complex the skin is as an organ. And all of these variabilities, as Elena already covered, will be reflected in the mRNA transcriptome. So for that reason, we really need a human skin cell atlas to capture all of these different variabilities. And again, just to put it in context, the anatomical variation of skin, prominent hair follicle growth on the scalp, and on our palms of hands. In fact, the skin can make the fingerprint ridges that are unique to each individual person. So skin is very fascinating. So why do we need the human skin cell atlas if you're still not convinced? So there are several different aspects that we're in particular interested in. One is to understand how all of these complexities and all of these cell types develop during embryonic development. And in fact, the fetal skin has some very interesting characteristics. One example of that is the fact that in contrast to the adult skin, that after wounding, it develops scar tissue. During embryonic life, if the skin is wounded before late second trimester, it has this incredible ability to heal itself without any scar tissue. The second important aspect of having a human skin cell atlas is that we really want to understand what are the mechanisms behind some very prevalent diseases, such as psoriasis and eczema. The third element is understanding mechanisms behind aging, and seeing really what's going on, what are the deriving factors, specifically in relation to skin. And lastly, having all of these information, we hope to be able to unlock new regenerative medicine avenues. And we will come back to this a few slides later when we cover a little bit more information. So with this and the background, we're moving to our second poll, skin specific.
Yeah, so again, so this is how many one, and it slightly scares me to think about this one a little bit. So yeah, how many skin cells does our skin shed per minute? Feels like this is almost a QI question for anyone who watches QI. So let me launch the poll there. So we've got an option of 3,000, 30,000, 300,000, or 3 million. So have a little think. Yeah, have a little think about how much you maybe want it to be or how much you actually think it will be. So yeah, put your answers in now. Again, just a reminder, if anyone's got any questions, just stick them in the Q&A bit, and when we get to the Q&A section, we'll make sure that we get to them there. Okay, if anyone hasn't put in an answer yet, do so now. If not, I will end the poll in 10 seconds. So yeah, if there's anyone still that wants to put in an answer that hasn't had a chance to do so now. Okay, so let's have a look and see what we've got. So the majority of the room are saying 30,000 skin cells shed per minute. That feels like a little bit like a tongue twister, that. 300,000 was the next highest. And then we've got a few people saying 3 million. So let's find out.
Cool. This time, I think everyone is now more familiar with our skin, so great. Yeah, so with this, let's move on to specifically the work that we've been doing in the Hanifa lab in relation to the skin, human skin. So here, we have been studying human developing skin from really early on during gestation until middle of second trimester almost. And in parallel to that, we are also profiling skin from across different ages to capture this effect of aging. And also we're looking at healthy skin and disease skin to better understand the mechanisms of disease. And I'm going to shortly show an example of one of our previously published work, which combines a lot of these elements, and we actually reported a really cool finding. So let's see what that is. So in this study that we published in 2021, in this study that we published in 2021, we profiled healthy human skin as well as disease skin. And specifically, it was eczema and psoriasis skin. And the healthy skin, just to clarify, so it was both adult and embryonic skin. And we profiled them using the same technology that Elena described. So single cell mRNA sequencing, and we built a human skin cell atlas with all of those different cells. And what we found was that it was incredible to see some of the cells that are present during normal development of the skin during embryonic life. They reappear and they start interacting with each other in the context of diseases during adulthood. And these diseases, again, they were eczema and psoriasis, but these were lacking in the healthy adult skin tissue. Yeah, this was a very novel finding, and it really captured our attention to focus more on the developing skin for the next work that I'll be covering. So in the next study, focusing on human developing skin, we built specifically an atlas of the cells of a wider time span of the development compared to the previous study that I mentioned. We had quite a lot of cells. And what did we actually do with all of these cells? So we actually were able to answer this key question. What are the cells involved in scarless wound healing during embryonic development? And we found out that these were some macrophages that are a type of immune cells, specific types of macrophages and specific types of skin fibroblasts were involved in this incredible tissue regeneration, which could potentially have significant impact. So if we can remodel this in adult tissue, it means that we can also regain this capacity to heal our wounds without any scarring. So now let's look at what are the things that we can learn from this developing human skin atlas. Imagine, all these different findings that we report and all these finely tuned mechanisms that give rise to a fully functional human skin, healthy human skin during development. And if we summarize all of that into a recipe book and say, okay, this is how we build skin from scratch, then can we actually use this recipe of human skin development to build skin tissue in the lab to kind of mimic that development of the human skin? And for that, we rely on these magical cells, I call them. So these are stem cells, and stem cells can turn into any cell type in the human body. So if we use the recipe that we've kind of put together and discovered, and kind of apply that to stem cells, then we would be able to skin tissue grown in the lab. But something to highlight is that this protocol isn't as simple as it looks here. It takes 5 months for the fully functional skin tissue to develop. But the striking thing is that it develops hair follicles. In addition to that, it very closely recapitulates all the different structures that we expect to see in the human skin, including the 3 main layers, as well as hair follicles, as I mentioned. We have nerve cells that kind of act as, they can sense the touch on our skin, and also glands, which is really impressive. And this is just to show you a real picture of these organoids. So this is how they look like when they're living their life in a dish in the lab. And we can envision how these lab-grown skin tissues can revolutionize clinical applications moving forward. So they can be used for skin and hair transplantation. And specifically, skin transplantation is one of the challenging problems in the dermatology field at the moment, because we know that in the context of specific diseases like diabetic foot ulcers, patients rely on skin transplantation. or people suffering from severe skin damage in unfortunate cases of burn, for example. And we can also model a whole host of different diseases by modifying how we're developing these organoids. And this will allow us to be able to test a lot of new drugs and see how the skin responds without using any animal models or actual human tissue. So this is very valuable for us. And if you really want to find out more about these skin organoids and stem cells, please stay tuned for the next Genomics Lite seminar delivered by our colleagues. And it's going to be the main focus of that talk. So just to put it all together, we can use Human Cell Atlas to understand diseases, how they develop, and also to hopefully improve how we monitor the disease, how we diagnose the diseases, and how we can develop new treatments for treating the diseases. And we can also use the Human Cell Atlas to optimize our in vitro tissue models, which we call them organoids. So these organoids, as I have already alluded to, they have multiple different uses, but one of the key uses for research specifically is to study human development without using the actual human tissue from the embryo, and also not to rely on animal models either. And so the next webinar on this, continuing this talk, is going to be on 25th of April. So yeah, with this, I'll hand over back to Elena.
Thank you, Feri. So I'll now give another future perspective, which is spatial transcriptomics. And basically what I've been talking about and what Feri has mentioned is single-cell RNA sequencing. However, single-cell RNA sequencing is kind of a way to measure single cells in suspension. And what you lose when you prepare the tissue for the single cell sequencing, you kind of lose the context of where those single cells were located within the tissue. So you lose all the spatial information that might be important, especially in the skin. You have like different layers, like the outer layer, the keratinocytes, the fibroblast layer, all located in different areas. And this spatial information is quite important as well. So recent technologies have now basically enabled us to do spatial transcriptomics, where we are kind of having a microscopy slide that has barcodes on the slide. Then you put your tissue on top. The RNA from the tissue binds those barcodes. And then when you remove the RNA with the barcodes, you kind of get the spatial information, because you can reconstruct where the barcodes came from that bound these RNA molecules. And this technology now enables us to kind of map the single-cell RNA sequencing results spatially. So we can generate like a whole map for the human body. And this was already done here in this collaborative work with the Bayraktar lab, where you can see how, for example, the hand develops. And you can see here, for example, chondroprogenitor cells appearing over time. And the idea is to use the spatial information similar so that we have a human cell atlas that is similar like Google Maps, where you have like a broad overview, like the country. Then you can zoom into the details, like look at the different streets in Cambridge, look at the garden of your neighbor if you want to. And then you can also zoom out and see the bigger picture. And in the sense of spatial transcriptomics and single cell transcriptomics, this would be like kind of you get the overview UMAP, like how the cells are connected with each other. You can zoom into each individual cell, see what is the gene expression of that particular cell, but you can also see the whole thing in the spatial context. You can see like where was the cell and how is it connected to other cells and to other tissues. And we basically have developed together with the Bayraktar lab a web atlas. This was done by Dave and Daniela in our lab and the Bayraktar lab, where you can already kind of zoom into the data and look at it yourself. You can kind of ask what genes do we find in which part of the developing human, You can see how frequent are particular cells, where are they located, and you can also see what genes is each individual cell expressing. And if you want, you can play around with this after this webinar. It's all online and accessible. And the idea is to use all this data and to make it accessible to everyone. So this is basically the concept of the Human Cell Atlas, to generate data that helps not only the scientists, but also the clinicians and the public to understand the human body better. And with this, we thank you very much for your attention. And if you have any questions, I think we still have a couple of minutes for questions. So maybe we can start some discussions now.
Yeah, amazing. Thank you both so much for that. It was really, really interesting. And Feri teed me up beautifully. This is wonderfully part one of a series of talks. So we've got part two happening in April. And as Feri said, we'll be looking at stem cells in a bit more detail. So I've just put the link in the chat to everybody on how you sign up to those. So if anybody has any trouble, just let me know. I will also send it around in an email. I don't know if it's also if we'll also get the link to the web atlas thing that you just showed, and I'll share that around as well. But yeah, we've got a couple of minutes still to do a bit of Q&A. So yeah, let's just get started with some of the questions that are in the Q&A bit and get going. So we've got one that says, when looking at healthy and diseased and across obviously aging as well, are you using the same individuals who are related and / or completely unrelated individuals?
Yeah. So we are using completely unrelated individuals, since it's very hard to sample the same person like during different ages. So if you would imagine you want to compare like a pediatric skin sample from a patient age 6 to an elderly patient age 70, this would be a study that would take too long to kind of look at the same patient. And also we get our samples, they are not taken specifically just for scientific purposes. So they are taken during surgery, and they're kind of like left over from surgeries. So we wouldn't like invite a person in and take their skin sample. So what we get is mostly just like when somebody has a surgery anyways, the bits and bobs that are left over.
Okay. And in terms of, you mentioned the humans. I've got a couple of questions in terms of the Human Cell Atlas is obviously super global in terms of all of those different all of the different sort of research labs in different countries and everything. Do your samples that you guys sort of work on in that regard, did they come internationally, or are they predominantly UK based samples that you were saying that potentially are left over from, say, surgeries or stuff like that?
So the samples that we receive, especially, for example, embryonic tissues, there is a specific resource. So HDBR, for example, Human Developmental Biology Resource, and that is a UK-based resource. But obviously, as I mentioned, it is also, for us, it's really important to capture, data from everywhere, all the different ethnicities. And obviously, we don't want to limit ourselves to just the UK. But for that, we don't necessarily, that's one of the benefits of having a global initiative. So you don't necessarily need to rely on getting the tissue yourself, then generate the data, then analyze. The data could be out there, generated by someone else in another country with the exact same method. So it allows you to then go and explore the data and kind of extrapolate the findings that we hope to achieve.
Yeah, amazing. And it kind of, so that kind of follows on a little bit to my second question about the globalness, is in the sense of, we've spent a lot of today talking about skin specifically. It's not just your team looking at skin, there are other teams looking at this around the globe, and it's all getting compiled together. Is that right?
Yes, yeah. Exactly. That's the idea of the Human Cell Atlas, that basically a lot of people are contributing to this massive project and sharing the data and making it accessible to everyone.
Amazing. Okay, another question that we've got through the Q&A button. If there's any more, do keep chucking them in and we'll get to them. So just wondering how you determine acceptable ranges for cell frequency and number of each type. Is there a different range associated with each pathology?
Could you repeat that question? I didn't get it.
Yeah, let me repeat again. I'll do it as slowly as I can as well. Just wondering, how do you determine the acceptable ranges for cell frequency and number of each type? Is there a different range associated with each pathology?
Yeah, I think what the question alludes to is that in the tissue, we find some cell types that are super rare, where maybe you are sequencing 200,000 cells, but you only find 10 of this particular cell type. And it depends on how different this cell type is from the other cell types. Like maybe it's just like looking very similar to another cell type, and then maybe it's not that relevant and you would just like group it together. There's no absolute value as to like just 10 cells don't make a separate group of cells. This is basically something that you decide based on how the data looks.
Okay,
Okay,
amazing.
something to that?
I think no, I think that was very well put, but also we now have techniques that allow us to enrich, if we expect, for example, a certain cell type is very rare, to be able to and if we know we're interested in that, we we isolate those cells before sequencing and kind of make sure that we have definitely captured those cells so that we know what to expect in the data later on as well. So there are some methods that we can apply.
Yeah, and basically the more cells you're sequencing, the more likely you're able to identify like rare subtypes. So that's also why it's so important to like combine lots of data and see if maybe you can find some new cell types that were just like grouped out because it was just 2 of them or something like this.
Okay, amazing. We've got another one that's just sort of, and I think this touches upon a little bit what you were saying, Feri, around obviously there's hormonal aspects of different skin sort of types and cell types in that regard. So is this captured in the data that you have around in the sort of analysis that is captured?
Yeah, so we have we usually, obviously we're limited to some extent, as Elena mentioned, by the tissue that basically is available, but we plan the studies in a way to make sure that we capture samples and tissues from different sexes. So yes.
Okay.
And we're also, for example, currently working on generating a pediatric skin atlas where you can see like hormonal changes during time affecting the skin. So I think this is something we're super interested in.
Okay, amazing. I'm conscious of the time, and we managed to get through a little bit of question and answer there. So that was great. So before we end, what I want to do is give a massive thank you to both you, Feri, and Elena, for spending the afternoon and into the early evening with us. It was a fantastic talk, and as it doesn't stop there, as we said, we've got part 2 coming up in April, so that's on the 25th of April, same time, same sort of place. We'll be in this Zoom webinar space, so do sign up to that if you are interested. Yeah, it would be oh, there's a really great question. I really don't want to miss this, which is just would you have any advice for interested undergraduates to start their career in similar fields? That's a perfect question to end on. So yeah, is there any advice that you'd give?
I can start by saying it's actually a really, really good sign that you know what you might be interested in, and definitely go explore possible options. If you're interested in analysis, maybe look for internships, placements, work experiences, on that side, or wet lab as well. So and I think the more broader areas in during that time someone can explore and be familiar with how things might feel like in different research fields, not necessarily just stem cells or what we presented today, and gauge your interest to know what you might want to do later on. Yeah, that's, I think, the advice.
And just ask a lab you might be interested in, or an institute you might be interested in, to join for a week or something like this to get an insight into what they're doing, because most people are super happy to show you what they're doing.
That was a really good point. Never be scared to reach out. I mean, worst case, people can say no, but
that sounds like a perfect message to end on. So yeah, once again, thank you both, Elena and Feri, for spending your time, and thank you to everybody watching for spending your afternoon and evening with us. Do sign up to the next month's event. This has been recorded, so I will send around a link to the recording just in sort of a couple of days' time. So you'll be able to watch back any bits that you missed. So yeah, without further ado, I just want to say, yeah, thank you again, and have a lovely rest of your evenings.